So, synapses. This is a word we have been using a lot, but we haven't actually talked about it in kind of a technical way. So today, don't worry, we are going to fix that. We are going to talk about synapses in a very technical way. To start, what is a synapse? We've already defined it. It's just the junction between a synaptic terminal, so the end of an axon, and some other cell. In this picture, it is one neuron and a second neuron, but as we'll see next time when we talk about muscles, it can be between a neuron and a muscle, and it can be between other cells. It's just the junction between a neuron and some other cell. There are two types of synapses. There's one that we're going to cover in this one slide, and then we're never going to see it again, and that is an electrical synapse. This is much less common. The idea with an electrical synapse is that it is a direct connection between the cytoplasms of adjacent cells. So think back to when we talked about plants. We talked about plasmodesmata, those little tunnels that connect the cytoplasm across the membranes and cell walls. Electrical synapses work in largely the same way. So you have a ring of proteins that can be closed or open, and these rings span across two plasma membranes. So you have the cytoplasm of one cell down here, you have a cytoplasm of the next cell up here, and when these channels are open, if this cell in the bottom experiences an action potential, the ions can just diffuse straight through those channels and trigger a depolarization in the connecting cell. So you don't have any extracellular transmission step in an electrical synapse. These are very fast. They're also a lot less common. So we're not going to worry about electrical synapses. For the rest of today, we are going to talk about chemical synapses. And right now, we'll cover what they are and how they work. Unlike electrical synapses, chemical synapses have a step that involves chemical transmission outside of the cytoplasm. And those chemical transmitters are called neurotransmitters. And we can see those right here. The little red dots are the neurotransmitters. Most synapses in the body are chemical synapses. There are three major structural parts to a synapse. You have the presynaptic cell right here. So this is the axon terminal of the cell that is sending the signal. You have a gap between the two cells that's called a synaptic cleft. And as I've mentioned previously, the gap in a synapse is extremely small. Think back to when we talked about the different types of intercellular communication. So this is going to be a gap of about 20 nanometers, which is extremely tiny. And that's important because this type of transmission is extremely rapid, and it has to be extremely rapid. As we'll see, it takes place via diffusion, so the only way for diffusion to happen fast enough is to have the gap be very small. And think about the delay when you think about wanting your arm to move to do a certain thing. There's no delay that you perceive, even though that process involves several chemical synapses. So that gives you an idea of just how fast the diffusion across this synaptic cleft is, and that's due to it being so narrow. And then third, we have the postsynaptic cell. That's the cell that's going to receive the signal and potentially transmit it onwards in the form of a new action potential. We're now going to go through this process step by step, breaking it up by different kind of parts. So we'll look at the presynaptic neuron first, and then we'll go through the rest of the steps. The first thing that has to happen before the cell is even going to transmit everything is you need to synthesize neurotransmitters. Neurotransmitters are going to be made all the way up in the cell body. That's where the nucleus is, that's where the ribosomes are, so the neurotransmitters are made all the way up here, and it can be really far away. They are then stored in synaptic vesicles. So here we have these little vesicles for storage that contain neurotransmitters. Those are called synaptic vesicles. These vesicles are synthesized all the way up in the cell body, and then they're transported down the length of the axon. The way they do this is actually really cool. They're little proteins. You don't have to know this, but they're little proteins that have kind of two active sites. They're enzymes that do two sets of things. On one side, they just grab onto a vesicle, a synaptic vesicle. And on the other side, they have little feet, and they walk along the cytoskeleton, basically. Each step requires a single ATP. So you have long fibers of the cytoskeleton that span the length of that axon, and you have proteins that are moving down the length of that axon, basically carrying a vesicle of neurotransmitters with them. And that's how the vesicles get from the cell body to the axon terminal. You don't need to know that. Don't worry about it. I just think it's cool. That all happens before an action potential occurs. Those synaptic vesicles are ready to go. Once an action potential happens, and the red arrow here indicates an action potential. Once that action potential reaches the axon terminal, that triggers, that depolarization from the AP triggers the opening of voltage-gated calcium channels. So this is a new type of voltage-gated channel that we haven't seen yet. Typically in your body, there is more calcium outside the cells than inside. So you open up these voltage-gated calcium channels, and that causes some of the synaptic vesicles to fuse, releasing their neurotransmitters via exocytosis. This is really interesting because this is not the first time we've seen a mechanism like this where calcium is causing vesicles to fuse, triggering exocytosis. Where have we seen something like this before? Take a second. Okay, did you get it? Where have we seen this before? We saw this before in the cortical reaction during fertilization. Remember, the sperm triggers a wave of calcium, and that causes the cortical granules to fuse with the plasma membrane of the secondary oocyte. So in the same way here, it's the same basic mechanism where you have an influx of calcium that is causing vesicles very close to the plasma membrane to fuse with the plasma membrane and release their contents via exocytosis. We saw that during fertilization with the cortical reaction, and we see it in synaptic transmission with the release of neurotransmitters. I always think that's cool that we have these repeated processes, the same basic mechanisms being used in very different contexts. So we have released our neurotransmitters. As I said, those neurotransmitters are going to diffuse across that synaptic cleft, across that little gap. And once they get across, they're going to bind to and activate very specific postsynaptic receptors. And I want to emphasize how specific these receptors are. There are many different neurotransmitters. You don't have to know any of them, but there are lots of different neurotransmitters, and they all have their own specific receptors. The binding of the neurotransmitter to the postsynaptic receptor results in something called a postsynaptic potential. A postsynaptic potential is a change in the membrane potential of postsynaptic cells. So we previously, in the presynaptic cell, we had an action potential, so we were changing the membrane potential of that cell. We then converted that signal to a chemical signal in the form of neurotransmitters. On the receiving cell, the postsynaptic cell, those neurotransmitters then trigger an additional change in membrane potential, and we call that a postsynaptic potential. This works because the receptors that are bound by the neurotransmitters are called ligand-gated ion channels. Ligand-gated ion channels. Ligand just refers to something that binds to something else. So anything that binds to another thing, we call that thing a ligand. So like, for example, when we talked about the endocrine system, when we looked at the water-soluble hormone transport pathway, the hormones that bound to the G protein coupled receptors, they are ligands for those receptors. So here, for neurotransmitters and synapses, the receptors are ligand-gated ion channels, which means that instead of opening from a change in membrane potential like a voltage-gated channel, these ligand-gated ion channels are open when some signaling molecule binds to it. So here we have the channels closed, but when the neurotransmitter binds to it, that channel opens up and it allows ions to diffuse through. In this case, these ions are going to diffuse through and cause a depolarization of that postsynaptic membrane. But I should clarify, because it doesn't have to depolarize that membrane. I'm saying that a depolarization results because we're talking about transmitting action potentials, and in order to do that, you need to depolarize your cell. But there are actually two types of postsynaptic potentials. There are excitatory postsynaptic potentials, or EPSPs. These are situations where that postsynaptic membrane depolarizes, so that brings it closer to a potential action potential. And you can see that right here. You have a depolarization in this figure. That is an excitatory postsynaptic potential. But you can also have an inhibitory postsynaptic potential, or IPSP. This is going to hyperpolarize that postsynaptic membrane, moving the membrane potential further from the threshold. And we can see that right here. So we have an I for inhibitory, and that membrane hyperpolarizes as a result of that IPSP. Now, what we're going to get into here is how signals like this can interact, because a neuron is not just receiving one signal at a time. It's being depolarized and hyperpolarized simultaneously from many axons at the same time. This process of having lots of signals combined together is called summation, and every cell that is receiving signals from synapses is constantly engaging in the summation of the postsynaptic potentials. Here is a micrograph of the cell body of a postsynaptic neuron with hundreds of synaptic terminals associated with it. These are sometimes going to send signals that amplify each other, and sometimes the signals are going to be antagonistic to each other, and the receiving cell is going to have to make sense of all of those conflicting signals. The resulting net postsynaptic potential is going to be a product of three main things. One is the interactions between the different signals, so some of them are going to be excitatory and some of them are going to be inhibitory, and that mix is going to indicate what the net signal is. Another factor that needs to be considered is the distance of a specific axon terminal to the axon hillock of the receiving cell. We talked about this a little bit last time, that the place where the action potential starts is the axon hillock. So in terms of meeting the threshold potential, it doesn't really matter what's going on in the cell body of the postsynaptic neuron. What matters is what's going on at that axon hillock. So the closer an axon terminal is to that axon hillock, it's going to have a greater effect. And then finally, we need to consider the concentration of neurotransmitter that is being used at each axon terminal. If there's more neurotransmitter, you're going to stimulate more ligand-gated channels generally, which means you're going to have a stronger signal, whether that is excitatory or inhibitory. So these three things together are going to dictate what the net effect of all of the different signals are going to be in a postsynaptic cell. There are two ways that postsynaptic signals can interact with each other. The first is temporal summation, which just means that you have two signals overlapping in time, temporal meaning time. So here's where you have, for a single synapse, for a single connection between two cells, you have two EPSPs arriving in rapid succession. The idea here is you have two signals in rapid succession, where the second one arrives before the first one dissipates, and we can show how this is going to interact graphically. So in this left figure right here, we have two signals that are arriving some distance apart from each other in time. This scale here is milliseconds, so it's not a lot of time difference, but it's enough time for the first depolarization to dissipate before the second signal arrives. In this situation, no action potential results because neither EPSP on its own is sufficient to hit that threshold potential. So we have signal 1, depolarization, it resets, then signal 2, and it resets. But in temporal summation, the second signal arrives before the first signal dissipates. So we have a depolarization, it starts to reset, we have a subsequent depolarization, and those two signals stacked together are stronger than either of them are alone. This leads to that postsynaptic membrane hitting the threshold potential, and we trigger an action potential in that postsynaptic cell. That's temporal summation. The second type of summation is called spatial summation. This involves two different axon terminals receiving signals at the same time, and those signals are going to overlap with each other. So we're dealing with different synapses associated with the same postsynaptic cell. If each of those synapses receives an EPSP simultaneously, that's going to lead to a stronger depolarization. And this also applies to IPSPs. So again, we can look at this in a figure. Here we have two EPSPs arriving simultaneously. So in this cell we have E1 and E2, so two excitatory signals arriving at the same time. Alone, neither of these signals is going to be sufficient to trigger an action potential, but together they can stack, and then you can hit that threshold potential and trigger an action potential. You can also have spatial summation involving IPSPs, either two or more IPSPs or a mix of excitatory and inhibitory signals. So in this figure on the right here, you can see we have an excitatory signal and we have an inhibitory signal, And then together, we could see how their combined effect is a weaker depolarization, because the magnitude of the original depolarization was greater than the magnitude of the hyperpolarization. So if you add them together, you have one signal coming from here, and another signal that is antagonistic coming from down here. You put them together and you have decreased the magnitude of that depolarization. It's still a net depolarization, but it's weaker, and therefore it's less likely to interact with other depolarizations to trigger an action potential. So that's spatial summation, where it's two signals coming from different synapses targeting the same cell, and those signals arrive at the same time and they interact with each other in some way. Either they stack with each other or they're antagonistic and they kind of cancel each other out. All of these signals are going to interact and come together at the axon hillock, which is the neuron's integration system. This is where all of the inputs from all of the presynaptic cells are going to come together within that postsynaptic cell, and we're going to figure out what the net effect is for a particular moment in time. Whatever the membrane potential is at that axon hillock, that is the summed effect of all of the EPSPs and IPSPs that cell is receiving at any given time. And remember, in the previous figures that we looked at, we were looking at two signals arriving at the same time. In real life, we're looking at thousands of signals arriving at the same time. So it's a very complex series of interactions involving those factors I talked about a few minutes ago. The big idea is that if the net membrane potential at the axon hillock reaches the threshold potential, then that triggers an AP in that postsynaptic cell, and the same process we just talked about is going to repeat at the other end of that cell. If you don't reach that threshold potential, then you will not trigger an action potential, because as we talked about last time, action potentials are yes or no. You can't go halfway. So that is the process of how synapses work, specifically chemical synapses.